So now our next step is the alignment. So first we check the quality of the reads. We noticed that there was a bit of a problem in the ends of the reads, so we trimmed the bad bits off. And now we are happy with the reads and we want to match them to the genome to figure out from where, which gene they are coming from. So we are talking about mapping and alignment interchangeably. Now, computers are very good at matching text, essentially, but here, and to make exact matching, but here there are some things that make this matching a bit more challenging to the programs. So first of all, the matches are most probably not going to be exact because, well, individuals have, of course, variants in their genome. On the other hand, also the reads have sequencing errors in them. And then also in genome, you have actually places which are not unique, meaning that the several sequences can appear in many parts of the genome. So then when you're trying to place your read somewhere, and if it comes from this sort of area, it's very difficult to say where it's really coming from. So these problems listed up here apply to any sequencing data, but then when we have RNA-seq data, we have one more problem. And this comes from the fact that when we are sequencing RNA, we are sequencing, of course, the mature RNA, meaning that the introns have been spliced away already. However, then we take these reads and try to match them to the genome where the introns are present. So inevitably, there are reads which, some of the reads which will actually go across exon-exon junctions. So the aligner program has to be able to cope with those situations. So if we look down here, so in my drawing, the green stuff here is a small part of a genome, of a gene. And it has an exon here and another exon here. And then it has a long intron in between. Then it produced some RNA. We extracted the RNA and sequenced it, and we have little reads, which are the black bits here, or these little sticks. And some of the reads, if they come totally within an exon, then they will of course match happily. But then some reads, like the purple one here, actually matches so that the beginning part of it matches to the first exon, and then the last part of it matches to the second exon. So somehow rather the aligner program has to be able to place this correctly to figure out exactly where is this border. And this is the difficult thing, because this distance can be thousands of bases long. And on the other hand, well, there are some splice site signals, but they are very weak, so you cannot rely on that. So this is the big challenge, and this is why running these aligner programs for RNA-seq can take a lot of time. Right, and then, yeah, well, maybe one more point here is that, so we are aligning to the genome, and that's what people usually do if you have a genome available. The good thing is that, well, it allows you to find new genes and transcripts. And it's also in some ways actually easier than mapping reads just to transcriptome. Because when you map directly to transcriptome, then, as you know, one gene can have several transcript isoforms. And if the read comes from such an area which is common to all these different isoforms, then it's difficult to say with any certainty from which isoform of that particular gene transcript it came from. So usually we map to genome. there are lots and lots of these mapping programs or aligner programs available. So we are using TopHat 2 here. There's another very good aligner program called STAR. This is something we are integrating into Chipster very soon. It's actually already in our development version, so we will be releasing it quite soon. And also maybe to note is that the people who made TopHat have also made another aligner recently called HiSat, which we might also integrate. And again, there is quite a good comparison paper where people have tried the different aligners from various aspects. But so today we are using TopHat, which is probably the most ever used aligner for RNA-seq data. And internally it uses another aligner which is called Bowtie. Now, one thing to mention also is until now, so Maria told about base quality, how confident we are in the base calls. Now I will talk about another quality value, which is called mapping quality. And this is now the confidence we have that the mapping position that the aligner program gives is a correct one. So this can obviously depend on many things, but probably the most important thing is how unique that particular sequence is in the genome. So if it's present in many places, then obviously we don't have any confidence whatsoever that it comes from one of them. And the mapping quality is calculated with the same formula as the base quality. So bigger the better. Some aligners do this slightly differently. TopHat has a kind of a fixed sort of set of numbers. So if TopHat tells you that the mapping quality is 0, it means that your read maps to 5 or more locations in the genome, and hence there is no confidence. Whereas if TopHat says that mapping quality is 50, then it means that it's unique mapping, and we are very sure that it comes from that place that the TopHat says. Then even if we are going to now talk about the TopHat aligner, I say a couple of words about another aligner called Bowtie 2, because TopHat uses this behind the scenes. And the thing to note here is that Bowtie 2 can run in 2 modes, but when it's automatically run via TopHat, it's always running in this end-to-end mode, which means that it requires that the read is aligned from the beginning to the end. So it doesn't tolerate many mismatches in the ends. It has another mode which would allow those ends to be just flipping around, but this is not the case when you run it with TopHat. So that means that actually TopHat doesn't tolerate well many mismatches. And so then very quickly we can go through the steps of the TopHat while your TopHat run is running. So if we start from here, so what happens is, so the red bits here are the reads, and what TopHat does for the very first thing is that if you supply it with the location of the known genes, exons and introns, it will actually use that information, and it will on the fly sort of create the transcripts from this information, because it knows the genomic sequence and it knows where the known genes are. It can construct from that the transcripts. So before it goes to the actual matching to the genome, it matches the reads to transcriptome, which it has made. And of course, many reads map at this point, which is good. So those locations are then converted back to genomic locations. However, some reads won't map, so these guys here, so they will go to the next step, and now the next step is matching the reads to the genome. And again, those reads that match nicely within an exon will happily map at this point, but some reads don't, and then they will go to the next step. And all these steps, this matching and this matching, actually happens with that bow tie aligner. So TopHat is kind of orchestrating the whole thing, but bow tie is doing the job. So then the ones that did not match are tried again, but in a different way, so that they are cut into pieces, into segments, which are by default 25 bases long. So then we try to match those 25 base thingies, and we looked for situations like this one here, where the first segment maps here, and the last segment maps here, and then the one in the middle, we don't know where it's supposed to go. So then this is a kind of easier now for the computer, because now it knows that, well, the middle bit has to go somewhere here. So it's a much smaller search space than looking through the whole genome. And so finally, hopefully, it manages to place it there correctly. Then TopHat will take a little bit of sequence from this end and from the other end and put these two pieces together like this. So it makes, this is called like a flanking database or splice side database. And then it takes those segments that did not match anywhere here. It tries again against these combinations and then hopefully manages to place the reads correctly. So at this point we would be basically done, but then it still goes back because now it knows exactly where these splice sites are. So it goes back to the, well, what we had in the previous slide when it was matching things to the genome. And there might have been situations where just one or two bases in the end did not match. So now it will try again because it has more knowledge, and hopefully it's able to then place those correctly as well. So you can imagine that now we have this very small data set. We have like roughly 100,000 reads, but typically you would have like 20 million. So running this would take a very long time. Then if we look at the parameters, so today we have single-end data. This parameter panel is now for paired end. So some things are the same, like the reference genome, which one do you want to use? But then some are different, like this parameter, expected inner distance between the paired reads. So this would be something that you would have to set according to your sequencing run, what they did for you in the sequencing facility. And the other important one to remember from here is this second parameter. So library type now refers to whether your data is made with a stranded protocol or not. And today our data is unstranded, but if you have stranded data, then it's very important to indicate it here. We have actually, this is a very confusing thing. People ask about this often, so we have made a whole manual page about this because there are different stranded protocols, and you have to set that particular parameter in TopHat according to the protocol that was used for your data. If you don't know, so usually you can get this information from the sequencing facility, but if for some reason you don't have this information for your data, then there's actually a tool in Chipster that allows you to check which protocol or which type of strandedness your data has. So there are lots of different kits, but what you want to see is, does it say stranded or directional or strand specific? And if it does has this, it means it's stranded, and you need to know in which way it is stranded. And then based on that information, you select the correct parameter. So either these parameters have a bit strange names, but they're either called first strand or second strand. And later on, when we do counting, we have exactly the same situation. So again, we have to know which way the data is stranded. Then another thing that can happen nowadays especially is people use a lot NextSeq, Illumina's NextSeq machine to produce RNA-seq data. And then you end up in a situation that if you have, well, for example, paired data, normally you would have two files. So one file containing the so-called read 1 reads, and in the second file the read 2 reads, and they are in the matching order. Now, if your data was made with this NextSeq, then it can happen that for one sample you actually get 8 files, so 4 pairs of files. So that's okay. But when you come to the alignment step, what you have to do first is to produce this file name list. So you give all the, so the endings are typically like this. So there is underscore and 1 and underscore and 2. So you would take all the underscore 1 FASTQ files and make a list, name list for those, and same for the underscore 2 files. And then it's this whole set that you give to TopHat later. And when your data is aligned, you get the result file, but now the format is different. It's not FASTQ anymore, it's BAM. And so what you have in the BAM, first you have a little header, like up here, where the first line tells if the alignments are sorted according to something in that file. In my case here, they are sorted according to the chromosomal coordinates. Then it will list all the chromosomes. So this was aligned against the human genome. So I have all the human chromosomes here and their lengths. And then the last header line is actually starts with @pg, which means program. So it tells which program was used to make this BAM file, and it will show the actual command here. So this can be handy if somebody gives you a BAM file. You can actually go and spy here how they made it. But after these header lines, then you start to get the real information, which is the alignment positions for each read. So like here I have the read name, then I have a thing called flag, and then it tells that, okay, this particular one maps to chromosome 1 to this position, and its mapping quality is 1. And then it has various other bits and pieces of information. Eventually it will have the actual read sequence and also the read quality string, the same that Maria was talking about earlier. So there are lots of fields and they always come in this same order. So if you want to know the mapping quality, for example, you will see that it's the 5th field on each line. There's also this thing called CIGAR, which may sound a bit strange. It certainly looks strange. It has numbers and letters. And the numbers are number of bases, and letters mean things like insertion or intron or so forth. So it just tells how well does that read map in that particular place. How does it map? Are there actually gaps in that mapping? And so then before. Let me see. Okay, it seems that, yeah, maybe we have an extra slide here, but anyway, so soon Maria will talk about other quality things, but I just mentioned this one. So once the reads are mapped or aligned, it's a good idea to check, well, how many of them mapped, how big percentage of the reads mapped, and of those that mapped, how many of them mapped uniquely, meaning in just one place. If they are paired reads, you want to know how many pairs mapped and how many of them mapped the right way, because they have to be in the right orientation with each other. And still about the file format, so you get a BAM file with the actual information of the locations, and you get another file which is an index file, BAI. This is just a little helper file that allows you to, or allows the tools like a genome browser to quickly retrieve alignments for a certain position. Because these files are very big, so if you had to go through them line by line, that would be easily 30 million lines, so it would take a lot of time. And once you have the BAM files, you can do various things with them, so you can check how many alignments, for example, it contains. And I have underlined the word alignments here because I want to remind you that one read can have and will have several alignments. And you can also get other statistics, and you can also make subsets for a certain region if you wanted to. And in addition to this BAM and the index file, you also, TopHat will give you another file which is called PED, which is just another file format. And PED files are typically used for giving information about some feature in the genome. It can be used for all sorts of features, but this particular one I have here as an example is a TopHat PED file which tells about exon-exon junctions. So it will have the name of the junction, where it is, and how many reads go across that junction. But so this can really contain different kinds of information, but there are always these columns. So chromosome, start position, end position, some sort of name, and some sort of score. So later on today we will have a PED file containing differentially expressed genes. So then our score is going to be the fold change of the expression. So you should have this four red boxes corresponding to the results. So you have the BAM file, which is the most important one, where the location information is. Then you have the index file, BAI, and the PED file with the junctions. And then you have the summary file, just the text file. And it's the summary file that I wanted you to have a look at this point. So, so it tells us here that our input was 86,568 reads. So that's how many reads we gave to the program. Of these, it managed to map, and this is the answer to the question 6, it managed to map 84,811. So the overall alignment percentage is very high, actually. It's 98%. And at this point, I should say that when you will be doing this with your own data, this is definitely going to be lower, but don't panic, it's fine. The reason why in my case here it's very high is that I had to make this small data set for you, and I wanted to make it in such a way that the reads come actually from one chromosome. It happens to be chromosome 19. So that means that in order for me to know that or pull out those reads, I actually had to align them already before, so these have been aligned, so hence it's not surprising that the percentage here is very high. And otherwise, so then in question 7, the question was, well, of these 84,000 and something reads, how many of these mapped to several places? In other words, they had multiple alignments. And there were 4,252 such reads. So I just wanted you to make a mental note for yourself here that definitely in the result file you will have more alignments that you had reads to start with, because some reads will have been aligned to many places. None of them were aligned to more than 20 places, but they were aligned to several. And I just want to show you quickly, let's have a quick, this was not in the exercises, but let's have a quick look at the BAM file as well. I will maximize this visualization window so that you can see it better. And now, just out of interest, so here we have the header. We can see that TopHat produces a BAM file which is coordinate sorted. And let's look at the very first alignment here. So here we have the read name. We can see that it aligns to chromosome 1 to this particular position. And then we can see that, hey, its mapping quality is lousy, it's 0. So that means that it maps to other places than this place as well. And then when we keep looking, so this is only so for every read there is one line, but now this is split into 2 lines here, so that we can see it all. But so if we continue looking further, so we have the sequence and we have the quality string for the bases, and then we get these tags. And one of these tags is NH, and it says NH is 7. So this NH tag actually tells that, yeah, this read maps to 7 places in the genome. So in our file here, this particular read will come up 7 times in different parts of the genome.